Melanoma Matters is an educational resource, and the opinions we express are our own and do not reflect medical advice. If you have questions specific to your own situation, please contact your own clinician. This podcast may be available in both audio and visual, so please check your platform.
Hi, everybody. Welcome to Melanoma Matters. I'm Sapna Patel.
I'm James Larkin. Hi.
We are going to talk about the Godfather 15 trial. Was it called Godfather 15 or just Godfather? Let me go back and look. This is the study of neutralizing GDF-15 antibody, overcoming PD-1 and anti-PD-L1 resistance in solid tumors. So this is a Nature paper published online December 11th, 2024. Ignacio Malero is the first author, Eugene Liao Sr., and it's called the Godfather trial is what it is. And this is essentially because we recently recorded another GDF-15 antibody, Consegramab, in cachexia that was published in NEJM. So that obviously got us to do a little bit of exploring on this molecule and so forth. Maybe before we do that, we'll do a quick fire intro. Let's talk about time travel. If you could teleport or time travel, James, anywhere in time, any era, or go back, any part of your life, redo, where would you teleport to? Let's say it's just a nice short stint.
Yeah, I don't think I'd want to go back and sort of try to redo any part of my life. That's done. It's boring, isn't it?
Yeah.
And I don't think I want to go too far back. For example, before the invention of general anesthesia, depending on how long you had to stay, because that kind of be nice having surgery if you needed it without general anesthesia. So I think maybe the 1920s, but of course, in the 1920s, lots of bad stuff was happening in the world post World War One. But I think the people in the 1920s who kind of had enough money were, from what one can gather, were really living a great life in lots of parts of the world. I think Europe and in the US actually. So, and I think that'd be fun. And it's kind of beyond living memory in a sense as well, I suppose. Actually, my grandparents were children in the 1920s. So they're dead now, but I don't think they'd have had particularly clear memories of it. So I think that's my answer.
You want to attend these parties like F. Scott Fitzgerald wrote about?
Flappers, or whatever they were called. Yeah.
Yes. What was that place called? West Egg?
Don't know. But yeah. Well, there's a certain sort of glamour there in a way as well, isn't there? Undeniably. And, all the clothes and the architecture as well and all that stuff. It's really pretty cool. And maybe post World War One, the people who did have money, that there just seemed so much joy then. Maybe because the war was behind them and over, and people were thinking, "Wow!" But I mean,
they didn't know what was to come, of course, as we never do. And
and also there's a massive amount of poverty, obviously, in lots of parts of the world in the 1920s as well. So you can't dismiss that. But this is just a sort of fun answer, anyway, isn't it?
Yeah, I mean, I probably would have a long time ago said Elizabethan era would have been my era, but also I would have wanted to be one of the high court ladies, not a lady in waiting, not I would have wanted to be. But I used to love all that Philippa Gregory novels about this period of time. I love like Queen Elizabeth I, all of this kind of stuff. So I would have said that. But recently, because I've gotten into watching Yellowstone, I absolutely would love to be just in the not the Wild West, as it were, but probably just the sort of ranching West before it was so commercialized, and just the beautiful life on horses. Life on ranches, that kind of stuff, where your
So,
anesthesia was just the bottle of moonshine.
yeah, tough life that though, undeniably. And also, when all those in the Elizabethan era, weren't all those outfits really uncomfortable as well? I mean, did take hours to get dressed and stuff like that.
Yeah, but what else did you have to do all day anyway? And you have somebody just like dressing you and tightening you and cinching you and
Well,
making your
yeah,
milk bath for you and putting, yeah, nice. But in the I'm gritty enough, I think, to enjoy horseback and not showering for a week or so and just figuring shit out. I think I would.
yeah. Well, there's no doubt a certain appeal to that.
I will say there's another part of me that would like to teleport back to your first days at the Marsden when Martin is teaching you and calming you down, as you said. Maybe I would just like to be a fly on the wall. Maybe not an interactive part of it, but that's another time I'd like to teleport to.
Yeah, maybe we should talk about that another time. I'm not. I think, yeah. Well, the moments of humour were great, but I was very sort of wet behind the ears and stuff like that, and very sort of very keen and geeky. I still am in some ways, actually. Many people that know me would say. Anyway, so next obvious question is, what is your background?
Oh,
Is
yeah.
is it real?
It's real. Shall I open the window so you can see? Shall I get it to where you can see there's a gondola moving behind me?
You have to keep it still.
Yeah, there we go.
Yeah, see the gondolas moving.
Yeah. Yeah, this is Steamboat Springs, Colorado. This is Western Slope, Northern Colorado. Beautiful. They're famous for their champagne powder here, they call it. Nice small mountain town. It's a pretty small mountain as mountains go. I think Utah has the biggest resort in the US, ski resort, or biggest mountain base, but this one's pretty small as they go. But yeah, just here for a conference and it's
And we're
beautiful.
January 25 for the record. But yeah, snow looks pretty good. Okay, so let's maybe talk a little bit about this paper. It does link well with the ponsegromab, like you said, which was in NEJM around Christmas. In terms of
September TBD at this point.
yeah, that's true as well. I mean, the other thing, just before we get going, maybe to note is there's a lot of authors here. We mentioned Nacho Malero already. So this was a study done in Western Europe, actually, not in the UK, but quite a lot of friends and colleagues, certainly people that I know, Reinhardt, for example, Duma, a guy called Juan Jose Soto Castillo, who was just our fellow very
Oh,
recently. Yeah, we're involved. Actually, Tom Kajeski's on this as well, actually, for the record. So Andreas is on it, Andreas Schmidt. I mean, Lex Egamont is on
yeah,
it.
you're
I mean,
right.
it's one of those ones, the more you look, the more you see, if you see what I mean. But it's a big authorship list. And then this molecule it's coming out of a biotech company, which I think is based in Munich. That's maybe worth saying as well. So that might speak to where the trial has been done. So do you want to maybe just go through a little bit of the background and sort of methods and things like that, and then we can kind of get into the bit of detail about it?
Yeah, sure. So GDF-15, you said it in the ponsegromab, and I forgot already what it stands for. It stands for, did they tell us? Oh, here it is,
Growth differentiation factor 15.
page one of the article. Yes.
Yeah, crazy place to put it.
Crazy. First appearance that they spelled it out. So growth differentiation factor 15, I think we are learning, you and I just learned that it's part of the TGF beta, transforming growth factor beta superfamily. And essentially, its expression is tied to immune suppression. Maybe that's the simplest way to say it. And so companies are developing these neutralizing antibodies. I think ponsegromab itself was a type of neutralizing antibody. This one is, we didn't practice saying this one either. Visugromab. Does that sound
I
right?
think that's pretty good.
Visugromab.
Yeah.
ISUGROMAB, CTL002, visugromab. So a neutralizing GDF-15 antibody. By the way, is it redundant to call it an anti-antibody? Anti-GDF-15 antibody? I guess, as opposed to agonistic antibodies. The point being, we have an idea of what the molecule looks like, and it's as monotherapy or in combination with nivolumab in PD-1 experienced cancers, essentially as a last line therapy, if you will. This was a phase 1/2 study. So in the phase 1, they were doing kind of normal 3+3 dose escalation across 5 dose levels. And I was remarking to you that the dose levels look like this is a molecule that looks to be similar to CTLA-4, that is ipilimumab dose finding, as opposed to maybe fianlimab or cemiplimab, where it's bigger milligrams. This appears to be an IgG along the lines of ipilimumab, if you will. But I'm just saying. And it was across 5 dose levels, 3+3, and then they were allocating for 25 patients. So then they backfilled the top 3 dosing levels once they reached MTD. It turns out they went all the way through 20 milligrams because they didn't have any DLTs. And so then they just were backfilling the top 3. And then in phase 2, they picked out a couple of tumor types, non-small cell lung cancer, urothelial cancer, to further look, though you and I were commenting there may be some other signals in the phase 1 that are interesting. So what do you think? Is that enough background?
Yeah, I mean, there's some quite cool things in there as well. I mean, this isn't my area of expertise, but this idea that GDF-15 is important in feto maternal tolerance towards placental alloantigens. I always think that that's a nice bit of biology. Is there something else, if you like, that is used by the immune system?
So what you're essentially bringing up is that we have like an inherent control we can look. If we want to look at GDF-15 expression, we know the placenta has a lot of it because you're trying to immune suppress the placenta so as not reject the fetus. And that comes up helpful when we start to think of things like immunohistochemistry and so forth. Your control tissue is going to be placental, GDF-15 expression.
Yeah, and then maybe the kind of the key point here is that they looked in the Cancer Genome Atlas for types of cancer where GD15 might be relevant for immunosuppression, which is how a non-small cell lung cancer, urothelial cancer came up. I mean, it's important to say that, maybe when we get onto the results a little bit, they did actually treat some other patients. I think colorectal cancer and melanoma, and the comment with those is that there wasn't really any significant evidence of activity. And then I guess the other sort of relevant broader issue, which we talk about a lot and is fairly obvious, is that most types of cancer, you do see some efficacy for anti-PD-1 or anti-PD-L1, but actually, depending on how you look at it, probably the majority of patients won't benefit. And it's sort of one of the holy grails, isn't it, in this research over the last several years, to try and figure out rationally, mechanistically, why these drugs aren't working better and what are the mechanisms behind it, and then can that be targeted. So I think that's really beautiful in this paper. And then my last general comment probably is just going to be something I talk about all the time anyway, which is this whole business about defining refractory cohorts. And maybe now is a good time to do it. And they do spend a bit of time talking about their refractory cohorts. And I think effectively it's people who've had previous anti-PD-1 or anti-PD-L1 and haven't benefited from it. But my point here, and I make it all the time, is that you've got your truly refractory patients. That's one group of patients, right? But actually there's another group of patients who've had the therapy in the past, and maybe they've had some benefit from it actually in the past, but have subsequently progressed. So, at the risk of repetition, I think there's going to be some patients in there who may have partial sensitivity to later treatment. And we talked about this a few times. And the Tony Rebus, Jed Walchop paper. I think it's in Cancer Cell, which we've done. We'll need to fact check this, which episode it was. Really speaks to this very nicely, because they're not the same group of patients, primary refractory versus someone who'd had the treatment in the past, maybe had some benefit, we don't know, and then gets re-challenged. So what I'm really getting at here is that there is going to be some activity, say, for nivolumab in this group of patients. Probably not much, but there is going to be some. But they address this point quite nicely, I think, in the paper.
I agree with you. I like, maybe at some point we can expand our thoughts on whether we think these phase 1 studies need to narrow down which refractory population they're treating, or it's okay if they sort of shotgun it like this and separate it out later, knowing that when you do that, your cohorts are going to get even smaller. And would you maybe have been better off to predetermine? The second comment I would make is we've talked about it for some time, and maybe this is now even more impetus, like you say, because we've brought it up so many times. We will do a podcast on the SITC position paper about defining refractoriness. Olivier Michelin at ESMO 2024, in one of his education sessions that he did with Inga Marie Spain and Tony Rebus, he brought up beautifully how that paper is really helpful. But there is a gap in that paper. There's a gray area between primary and secondary resistance. So we will talk about that. And maybe when we do that paper, it will be the time for us to talk about, in melanoma, what would be our benchmark for primary refractory, never got a response to PD-1 based therapy. What's the benchmark for your next line of therapy? Metrics, PFS, objective response rate, versus if you were a secondary resistant. Much to your point, don't we have to bake in that somewhere between 10 to 15% of any response that you see may just be sensitivity to that checkpoint and not really the additional whatever you're adding to it. So it might be nice for us to think through. I mean, we're doing it just as an academic exercise for you and me, but just to think through what would be interesting in drug development in both of those spaces for melanoma. And then frankly, your thoughts on kidney cancer too, in that same regard. So I agree with you. Refractoriness definition is, it can sound like a broken record, but this is where the rubber meets the road. So.
Yeah, absolutely. I mean, I think just in this paper, I think what you said, I'm actually quite comfortable with it, if you want to call it a sort of scattergun type approach, because at the end of the day, you've got a pretty refractory population. But I think the real strength here is that, and the rational biology, those kind of orthogonally, if you see what I mean. So this is the biology, we're targeting it. We've actually gone and looked in these particular types of cancer. And in many ways, I think this is obviously just a first step, because the real question is going to be subsequently, how are you going to develop this, which groups of patients, because you're going to need to do randomized trials, obviously. And then that question will then come out in the wash anyway.
What I found really nice also is, based on biology, clinical need, but then they also went to verify that it was on target. Essentially, did we adjust the level of immune suppression in these tumors? So they have baseline biopsies, they have day 14 biopsies, which is after, in the at least dose escalation, it's after some of monotherapy. And then when they backfilled, they just went straight into combination. They have day 28 biopsies, which are after combination. And all of these are showing a change. So you're seeing, after you block this GDF-15 and sort of change this immune suppression, you get more T cells in the tumor, period. Now, what are the flavors of those T cells? Because ultimately the ratio of effector to regulatory T cells matters. It does still look like you're getting, perhaps a higher population of CD4 FOXP3 regulatory T cells on treatment. But look, the point being, they're at least showing, yes, blocking this does change the context of the tumor environment. Now they're going to have to nuance that a little bit more. Does coming in with more PD-1 blockade, does coming in with CTLA-4 help deplete some Tregs and then keep the ratios more favorable for effector T cells? I think it's pretty nice in a phase 1 to see biology, like proof of biology like that. And they did it in a good number of these patients. I wrote down the numbers, like 22 out of 25 of the patients had a baseline biopsy That's incredible, right? That's like 80 some percent, 88% capture rate, 13 of 16 on day 14, 17 out of 25 at day 28. That's great for a phase 1 trial, in my opinion.
Yeah, absolute kudos, I think, to the investigators, because this stuff isn't that easy to do. And obviously this is in nature, which I think reflects all of these things. Not just are we seeing some evidence of efficacy in a situation where you wouldn't really expect to see much with nivolumab on its own.
And are we seeing any efficacy?
Yeah, I mean, you're seeing sort of response rates with, I think, decent potential response durability in both of those cohorts are sort of around the, 10 to 15%, 20% type of mark. So I think that's definitely of interest. And the other thing that they did is they kind of compared, if you want, the quality of those responses to the benefit or otherwise people had had previously, and show that actually the responses were at least as good, if not potentially better than what people have had before. I mean, all of this is enough for me, to be quite honest. I mean, what one could sort of go through this with a fine-tooth comb if you want and be really picky about that. But I think at the end of the day, they've demonstrated what they set out to demonstrate. And the real question for me is, what you do next and how you would do, I think, randomized trials. I mean, I've seen enough here. We haven't talked about safety, but we can do that in a minute. But if it was me, I would be thinking immediately, and I said this when we talked about the ponsegromab. I'm still going to need a lot of tuition on how to pronounce that, I think. I was in areas of significant unmet medical need. I think as soon as you've got you're happy with the safety, as it were, I would really be going straight into randomized trials, actually, with, ideally pretty hard efficacy endpoints. But yeah, carry on.
Well, I think that's a very meaningful comment, because you and I have been sufficiently critical about companies who go straight from phase 1 to randomized, let's say, registrational trials. So, again, this whole podcast is an academic exercise for you and me, but for our own ears to hear that, like, this is enough. This is enough to say I'm sufficiently interested in these, certain groups. Would I do it in all tumor types? No. Might go a little bit further to see, does GDF-15 expression at baseline correspond with any of this response or correspond with any of the day 15? Did you actually get, upregulation of more GD15 by blocking it? Did you saturate all the GD15? There's a little bit more to do here to figure out things, but I agree with you. I think if you're happy with that response rate, this is enough. Now that's careful, because as I say, we've been, I think, critical in the past of people going too rapidly to a phase 3, but this one certainly looks good for a randomized, maybe even a phase 2. Jeff Weber's favorite design, as he told us. The other thing is, you said it's something about safety. So what I wrote down here is safe, basically safe and tolerable. The grade 3 treatment emergent adverse events, grade 3 in about half the study, 48%, but that's okay. I mean, I don't
Yeah, and also this is a sort of heavily pretreated cohort of patients, and it's relatively small numbers. And I think you just need to be a little bit cautious. I mean, and that, for me, that's part of the point of actually doing some sort of randomized trial. It's not just going to be about efficacy. It's going to be about safety as well. And I think the key reason that this is a little bit different, probably two actually, or maybe even three, that it's rational scientifically to do this. Okay. B, they've actually got correlative data embedded in the trial. And then 3, this whole business about comparing benefit within patients effectively down the line. So there's actually several reasons, I think, that this is pretty solid. The only other comment I was going to make actually is about the other molecules that are sort of out there at the moment, which is to say, I think there's an AstraZeneca compound as well that's being tested. And I think that so far, at least in terms of what's in the public domain, there hasn't really been evidence of efficacy in the same way that we've seen for this and the Pfizer drug. So I guess that's one thing. But again, this is all relatively early stuff, and there could be all sorts of reasons for that, I think. So I wouldn't necessarily read too much into that, I don't think.
In closing, maybe final thoughts. Would you run a GDF-15 antibody trial at the Marsden?
Yeah, I'd love to. Yeah, I'd love to. And actually, I've already talked to a colleague of mine about this the other day. Uh, might not say too much more, but who treats cancers where there is a really significant unmet need. And I may have alluded to this anyway when we talked about the Ponsegromab paper. And yeah, and we didn't mention it actually on this paper, but just to mention it again now because I forgot.
Yeah.
The patients who had the high serum levels at baseline gained weight exactly like in the other study. So that's very, very strongly suggestive and I think supportive as well. So I think particularly there's two bits to this, aren't there? Those cancers where cachexia is a big problem as per the other paper, but as well as this, you've now got some biology and suggestion that independent of that, probably it could help immune checkpoint inhibitors to work better. So yeah, we'd love to have a trial here, but
It's going to be
we'll
all
see.
vertically integrated. Checkpoint, GDF-15, weight gain, GLP-1 agonists for weight loss now. We're just going to be only joking. So I would enjoy a GDF-15 trial in Colorado. But what I would say is I would like to help with some of the biology because obviously I really only treat melanoma. And I don't know that we see the signal here, but I would caveat that some of the preclinical work was based off of the TCGA, which is primary and nodal, lymph nodes for melanoma. Maybe we could do some of this on some of the metastasis. Some of the transcriptomic data suggested for an ocular melanoma patient or two, maybe a correlation with GDF-15 and T cell infiltration, or GDF-15 and interferon gamma, for which there should be very low here. So maybe that's the correlation they're trying to make. Again, I would caveat that the uveal TCGA is the wrong place to look at this, but I think it would be very interesting just to look in our banks and just look at GDF-15 expression on some metastatic tumors or transcriptomic data, and then figure out exactly this idea about the sort of T cell population. Yeah, I feel the same. I'm very taken by this idea that it can potentially help with weight gain. And as you and I are somewhat obsessed with, then body composition is everything. It's not just about weight gain of, more pounds on the scale or kilos on the scale. It's, can it be proper protein creating muscle, creating a better immune sort of composition for the body. And anyway.
Yeah, and metabolic health as well, which I think is going to go hand in hand with that. So yeah, so it's great. So kudos to Nacho Malero, I think, and our friends and colleagues are involved in this, and we're going to see some more of this, aren't we, over the next few years.
Good. Well, thanks for this, then.
Happy snowboarding.
Thanks.